What if a T cell could fire off a full immune response without ever recognizing its actual antigen? Superantigens like Th1 skip the antigen groove entirely. They staple the TCR's V-beta domain directly onto MHC class II, switching on huge numbers of T cells at once and triggering a cytokine storm that causes toxic shock syndrome. In this video, we'll cover T-cell activation and its checkpoints, superantigens, the Th1/Th2/Th17 and Treg subsets, cytotoxic T cells, and how B cells turn all of this into a high-affinity antibody. And I'll flag the timestamps in the description below if you want to jump straight to one. Let's start where every adaptive immune response begins: how a naive T cell actually gets switched on. Naive T cells get activated in secondary lymphoid organs, and they need two separate signals before they'll commit. The first is antigen recognition, the T cell receptor binding a peptide sitting inside an MHC groove on an antigen-presenting cell. Which co-receptor is involved tells you which MHC class you're looking at. CD4 pairs with MHC class II, while CD8 pairs with MHC class I. Underneath all of that, the CD3 complex is what actually carries the signal into the cell. Without this antigen-specific signal 1, there's simply nothing for the T cell to respond to, which is exactly why a second, non-specific signal still has to happen before the cell fully activates. Once signal 1 is delivered, the T cell isn't done yet. It still needs a second, non-antigen checkpoint before it can fully commit. That's costimulation. B7-1 or B7-2 on the antigen-presenting cell binding CD28 on the T cell, and this B7 expression actually goes up during active infection or inflammation, which is what tells the immune system this is a real threat. If that second signal doesn't happen, the T cell doesn't just do nothing, it becomes anergic or tolerant, essentially shut off to that antigen going forward. When both signals do occur, the T cell makes its own IL-2 and drives its own proliferation. And here's the high-yield detail: CD8 T cells can't fully differentiate on their own. They depend on help from CD4 cells to get there. Now that costimulation switches activation on, let's look at the molecule that switches it back off. CTLA4, also called CD152, sits on activated T cells and on Tregs, and it competes directly with CD28 for the same B7 molecule, except binding CTLA4 delivers an inhibitory signal instead of an activating one. Block that break with a drug like ipilimumab and you release the immune system to attack tumors harder, which is why it's used in melanoma and colorectal cancer. Go the other direction and give a CTLA4 agonist like abatacept or belatacept, and you dampen T cell responses, which is useful in rheumatoid arthritis or to protect a transplanted kidney. And here's the pearl that shows how critical this break really is: CTLA4 knockout mice don't just get mild autoimmunity, they develop fatal lymphoproliferation. That checkpoint controls a normal, antigen-specific T cell response, but toxins like Th1 and the staphylococcal enterotoxins bypass the whole system. Instead of sitting in the peptide groove, a superantigen cross-links the TCR's V-beta domain straight onto MHC class II, outside where antigen normally binds. Because that connection doesn't depend on the T cell recognizing a specific antigen, huge numbers of unrelated T cell clones get switched on all at once. That drives a massive release of IFN gamma, which activates macrophages, and the result is a full cytokine storm of IL-1, IL-6, and TNF-alpha. Clinically, that's what produces toxic shock syndrome and this kind of toxin-mediated food poisoning. Once a T cell is fully activated, its next job, if it's a CD4 helper, is deciding which of three main personalities it's going to take on. Th1 cells are induced by IL-12 in response to intracellular pathogens. They pump out IFN gamma, and that cytokine both activates macrophages and drives IgG class switching. Th2 cells go the other direction. Helminths and allergens push naive cells toward IL-4, IL-5, IL-10, and IL-13, which switches antibodies toward IgA and IgA and recruits eosinophils. And Th17 cells need TGF-beta plus IL-6 together. They respond to extracellular bacteria and fungi and their signature cytokines, IL-17 and IL-22, recruit neutrophils, and reinforce barrier defenses. With those three profiles in mind, let's trace exactly how one naive helper cell commits to a single path. If the local environment is rich in IL-12 and IFN-gamma, the master transcription factor Tbet gets switched on and the cell becomes Th1. If instead IL-4 is present without IL-12, GATA3 takes over and pushes the cell toward Th2. And if the mix is TGF-beta together with IL-6, RORT drives differentiation into Th17. This exact fork shows up clinically in leprosy. Patients who mount a Th1 response get the tuberculoid form, with granulomas that keep the infection controlled, while patients who shift toward Th2 get the lepromatous form, disseminated disease that antibodies alone can't contain. Quick check before we move on. Which Th subset is induced by TGF-beta together with IL-6 and runs on the transcription factor RORT? Drop your answer in the comments, then keep watching to see if you got it. Building on what you just covered with those three helper subsets, there's a fourth CD4 lineage whose entire job is to calm things down instead of ramping them up. Tregs are defined by the marker set CD4 positive, CD25 positive, FOXP3 positive. They work by secreting IL-10 and TGF-beta, and those cytokines actively suppress Th1 and Th2 responses that would otherwise keep firing. That suppression isn't optional, it's essential because without functioning Tregs, you lose the brake on self-reactive T cells and autoimmunity follows. While Tregs are busy restraining the immune system, a completely different cell is doing the opposite, actively killing infected cells. CTLs are CD8 positive T cells, and they recognize antigen presented on MHC class I rather than class II. Just like helper cells, they need signal 1 plus costimulation, but full cytotoxic function also depends on cytokines from Th cells, especially IL-2. Once fully armed, their effector toolkit is perforin and granzyme released to punch through and destroy the target cell, the phosphosL pathway to trigger apoptosis directly, and IFN-gamma secretion on top of both. So far, every cell we've discussed has been a T cell, but B cells can actually skip the T cell step entirely for one specific category of antigen. Thymus-independent antigens are things like lipids, polysaccharides, and LPS. Because of their repetitive structure, they can cross-link B cell receptors directly, activating the B cell without any help from a T cell. This route is mainly handled by marginal zone B cells and B1 cells. But that speed comes at a cost. The response is mostly IgM, it's weak, and critically, it generates no memory at all. That fast, T-independent route works for certain antigens, but most B-cell responses actually need a T cells help to reach full strength. Thymus-dependent antigens are protein antigens, and they specifically require CD4 T cell help to get going. The B cell internalizes the antigen, presents it on MHC class II, and, in the process, upregulates B7 on its own surface. The key second signal here is CD40L on the T cell binding CD40 on the B cell. That's the licensing step that lets everything else happen. With both signals in place, the response builds into a germinal center reaction, complete with affinity maturation, isotype switching, and long-term memory. All of this plays out in two waves, an early wave of IgM-secreting plasma cells, followed by a slower germinal center wave that produces high-affinity, class-switched antibody. Once that germinal center reaction is underway, two more processes fine-tune the antibody response even further, starting with affinity maturation. Inside the germinal center, B cells undergo somatic hypermutation, which introduces point mutations specifically into the variable regions of the antibody gene. Because of this, B cells whose new mutations happen to bind antigen more tightly outcompete their neighbors for that limited antigen, which is clonal selection in action. Repeat that cycle enough times, and the average affinity of the antibody pool climbs steadily higher. Alongside that affinity fine-tuning, the antibody's entire class can change too. That's isotype switching. Mechanically, it's DNA recombination at the heavy chain constant region switch regions, and which class you end up with is driven by cytokines coming from helper T cells. There's an important catch. This is a one-way process. Once a B-cell has switched away from IgM, it can never go back. The specific cytokine tells you the destination class. IFN gamma drives switching to IgG for opsonization and complement fixation. IL-4 and IL-13 drive IgA for mast cell, basophil, and eosinophil responses, and TGF-beta together with IL-5 drives IgA for mucosal immunity. And here's the pearl that ties affinity maturation and isotype switching together. Switching classes actually lowers avidity because the Fc portion changes, but affinity maturation compensates by making each individual binding site stronger. So that's the complete two-cell story, from AT cells signal 1 and signal 2 all the way through to a class-switched, high-affinity antibody. Remember the breaks. CTLA4 and Tregs are what keep that activation from running away. Remember the flavors. Th1, Th2, and Th17 each answer a different kind of threat. And remember the payoff. A Thymus-dependent antigen, a germinal center, and 2 rounds of fine-tuning are what get you from a naive B-cell to a high-affinity, class-switched antibody. If this helped, drop a comment with your knowledge check answer, and I'll see you in the next one. Thanks for watching.